Alright, welcome to video 3 of module 7. Just a very quick reminder again, if you do want to sign up to the upcoming crash course, the link is down below and spots are limited, so I will take that link down once it fills up. Right, so we did go into different types of pathogens, the chain of infection, how pathogens can spread. And then in the second video, we went through how the immune system, plants and animals respond to different types of pathogens. Now we're into inquiry question 4, looking at how infectious diseases can be controlled. This part of the module, there's going to be a little bit less focus on content. Rather, there is going to be a lot of scientific and working scientifically questions where they give you graphs and you have to analyze the trends in the graph, or they give you diseases and you have to tell me what is going on. Is there a decreased incidence, increased prevalence, morbidity, mortality, those sorts of things. So more analysis, more data analysis, working scientifically sort of questions. Okay, a lot of dot points under this inquiry question, but they are all pretty simple, I promise you. Okay, so first of all, looking at a few factors that can impact local, regional, and global spread. Okay, local factors include personal hygiene, so washing your hands, putting on masks, making sure you dispose of waste thoroughly, okay? Not mixing knives when you are chopping chicken and fresh vegetables or fruit, okay? That's really important to prevent gastro diseases and public health responses, okay? So making sure that we just reduce the chance for host-to-host transmission. Regional factors include surveillance and area quarantining, okay? And global factors include global surveillance, okay? So some regional factors can include like whole area quarantining. Another thing is the pipelines, okay? Very common transmission in developing countries is fecal oral route, which means that pathogens in your feces enter your oral route. And you might think that's quite disgusting, but it does happen a lot in developing countries because their pipelines, we often have water contamination and they don't kind of keep their food very clean, which is why we get the Bali tummy. So, very common term there for travelers going to Bali or other countries like Thailand. Often, a couple days into your trip, you get a bout of vomiting and diarrhea. Why does that happen? Because of that fecal oral transmission or just bacteria not being thoroughly cleaned out of your food, or the food is not cooked to an adequate temperature, and your bodies are just not built for that. Whereas people in Thailand and Bali, they're used to it. Okay, they have developed immunity to those types of pathogens. If you stay in Bali for a couple years, I'm sure you will develop immunity to those pathogens as well. Right, so that is an example of local, regional, and global factors. Okay, um, let's go through a specific example just so you understand this a little bit better. Okay, local spread of influenza, very common flu, deals with local environments such as schools, hospitals, and suburbs. So making sure that it doesn't spread beyond those areas. And often schools and hospitals, they're very common sites for transmission, especially like preschools and kindergarten. You probably got sick like every week because kids are dirty and they don't keep their personal hygiene very well. Okay, and the spread of influenza regionally can occur through the use of airplanes and cars, so traveling to bigger areas, okay? And this can also lead to global spread like the pandemic of COVID-19. How do we prevent this spread? Okay, hygiene practices, the first and foremost, really, really important because if you get rid of the pathogens on your hands, very unlikely that you will get sick. Quarantine is also a really good one. I'm sure you guys all know what quarantine is by now from 2020, 2021. It's just staying at home, okay? Keeping sick people away from infected people. That prevents the spread of the pathogen from the infected person to the sick person. Public health campaigns, that's very important. Public health campaigns are funded by the government, okay, and that just minimizes the occurrence of disease in whatever they are trying to get you to do, whether that is immunization, screening programs, or healthcare like Medicare, that is a public health campaign because Medicare is funded by the government. Okay, a very good example is the COVID-19 vaccines. From memory, the Pfizer vaccine cost almost $100 each shot, but it was subsidized by the government. So that is a public health campaign that just increases uptake of vaccines. Because I bet if everyone had to pay $100 for that vaccine, no one would have gotten that vaccine. Okay, so it just increases the amount of people getting that vaccine, which is what the government wants, so that more people can work and there's more income for Australia. Okay, another thing is pesticides, really important for plant diseases. So that gets rid of mosquitoes, rats, pathogens, and other vectors from transmitting the pathogens to other organisms. Genetic engineering, that is an interesting one. Okay, we're looking at gene drives for mosquitoes carrying malaria. That is from Module 6, so go back to Module 6, review that if it sounds unfamiliar. But that is also a prevention method. Now, gene drives are not implemented very widely because there's a lot of ethical and social dilemmas that come with it. So that is probably a future direction for preventing pathogen spread. Right, moving on, a very big one is vaccinations. Okay, vaccinations, if you see a question on prevention, immediately I want you to go to vaccinations. Why? Because vaccinations are one of the most important things that we can do for preventing a disease. When COVID-19 came, the first thing that pharmaceutical companies thought of was vaccinations. Vaccinations have eradicated certain diseases in the world, such as smallpox. Smallpox is the first disease to be completely eradicated by vaccinations. So it is really, really important. There are some other diseases that are nearly eradicated, like polio, still a couple in developing countries, but we are nearly there. So it is very important. Okay, some key terms. Vaccination means having a vaccine. A vaccine is a substance given to stimulate the body's production of antibodies and memory cells. So we're trying to get the immune system to produce our memory B and T cells, just so we get that secondary immune response where we have a faster and more effective response when the body actually encounters the real pathogen. And this provides artificially acquired immunity. Okay, immunity means you are immune to a disease that could be through infection or it could be through vaccination. Okay, this also creates a development for herd immunity. Herd immunity is when a large population, a large proportion of the population is immune, and that just protects weakened people who have they can't take vaccines, whether they're elderly or very young, or they have immune systems that aren't working very well. Okay, so herd immunity is very important to protect sick people in the population. And we generally get herd immunity when around 90% of the population is vaccinated. Okay, go back to our notes to have a revision of herd immunity. These are different types of immunity. I definitely want to know the definitions of each. Okay, so active immunity is when your body naturally produces those memory B and T cells. Your immune system is working to produce those memory cells. This can be natural active immunity, which means you naturally get infected by that pathogen, or it could be artificial active immunity, which is when vaccinations stimulate the production of memory B and T cells. Passive immunity is when we don't have any production of memory B and T cells, but you are immune to that disease because you have antibodies. Antibodies provide you immunity that doesn't last very long, but it makes you immune to that disease in that moment of time. That can include natural passive immunity, that is through maternal antibodies. So babies, when they're really young, they don't have an immune system that can produce memory B and T cells very well. So how do they fight off diseases? It is through maternal antibodies, which is why after the birth of a baby, doctors really recommend females to have breastfeeding for their baby. That's because in breast milk there is so many antibodies within them. So the baby gets access to all of these antibodies and does build up their immune system as well. Um, Yep, so that's natural passive immunity. The other one is artificial passive immunity, is when you get injected antibodies. That can be through IV drips or intravenous injections. Not very commonly used. Okay, it's only for people who have immunocompromised immune systems and they can't produce their normal antibodies very well. So not used very commonly, but still used for some people. All right, some treatment strategies now. Okay, so how do we treat diseases? Okay, if we failed in preventing pathogens from entering our body, now what we're looking at is treating those diseases. We have two main things. Obviously, we have antifungals as well, but in the syllabus, we want you to remember antivirals and antibiotics. I'm sure you've heard about antibiotics before. I'm sure you've taken some. Antivirals not as commonly used. Okay. One thing I want to really highlight is the pathogens that each of them prevent from spreading. Okay, antivirals prevent the spread of viruses, as the name suggests. Okay, so they aim to inhibit viruses from replicating by attacking proteins of that virus. They can also prevent protein synthesis of viral RNA or DNA. Okay, so they don't attack the virus directly, but they inhibit and disrupt their life cycle. Okay, so they can actually prevent entry of the virus into our DNA, our nucleus. They can prevent the virus from leaving. They can inhibit translation factors. Okay, they can do a lot of things to inhibit the virus from replicating. Those are antivirals, only effective against viruses. Antivirals do come with some consequences. They do have some side effects like vomiting and diarrhea. And obviously, anti-viral resistance can be developed. Antibiotics, on the other hand, they treat bacterial infections. Okay, so don't take antibiotics just because you're sick. Okay, you have to know that you have a bacterial infection because antibiotics specifically treat bacterial infections. They either kill the bacteria or inhibit their metabolic functions, preventing them from spreading, or affect the transcription of proteins of bacterial cells. Okay, so they can either kill the bacteria directly, which is different to antivirals, which do not attack the virus directly. And the big problem of antibiotics is antibiotic resistance. Okay, if you take too many antibiotics or you don't take it properly, for example, you don't take the whole packet of antibiotics and you take one or two, antibiotic resistance can occur, which is when bacteria develops resistance against antibiotics and antibiotics can no longer kill them. Okay, so that is really, really dangerous because we have a lot of bacterial diseases where we can no longer treat with general antibiotics anymore. And we have to use very strong antibiotics. An example is golden staph in hospital situations. Very bad disease. All right, moving on. How do we control the spread of disease? Okay, we're not looking at treatment anymore. How do we control it? PPE is very important. So personal protective equipment, using masks, using gowns, using gloves, goggles, whatever you have to do to protect yourself to prevent entry of the disease and the pathogen. Contact tracing is quite important. I'm not going to go through it. You probably did it in COVID-19. Thermal scanners used in airports very commonly. They can detect people with a fever, and if they do have a fever, they put them into a separate little room, interrogate them, and re-quarantine them if they're infectious. Disinfectants, sanitizers, adequate ventilation, and quarantine. Very effective control methods. Now, for this part of the module, questions are most likely going to be data analysis questions and also getting you to analyze the effectiveness of each control method. To do that, you have to give me an example. So, for example, you can say quarantine is a very effective control method to prevent the spread of COVID-19 because blah, blah, blah. Okay, so tell me what it is and what it does. All right, interpreting data. Really important slide here. Considering the questions you're going to get for this part of the module is going to be data analysis. These words here, I want you to remember off by heart. Incidence is the number of new cases of disease. So if you see a graph showing you incidence, you can really tell the effectiveness of certain prevention methods. So for example, if a vaccination program was implemented in 2007, and before 2007 the incidence was really, really high, and then after 2007 we see a drop in incidence, we know that it is the vaccination that is causing this decrease. Obviously, correlation does not equal causation, but you can kind of draw connections between the data and what has been implemented. Prevalence is the proportion of people who have the disease at a given point in time. Okay, so this is different to insulin, where it's a number of new cases. So prevalence is looking at overall in the population, how many people have the disease. So it could be people who had the disease previously, they have not recovered yet, or it could be the number of new cases. It's all added together to add to prevalence. Mortality is the um, rate of a disease over the particular number of deaths due to a particular disease at a specific time period. Okay, so read over these definitions, understand them, and then go back to our notes as well and try to answer a few questions. Okay, there are a lot of data analysis questions here. Moving on, we're going to go through some specific diseases Malaria and dengue fever. Okay, these are the two diseases that I want you to remember one of. Okay, so the syllabus says malaria or dengue fever. So remember either malaria or dengue fever. The one that I like to remember is malaria, just makes more sense to me and makes more sense to a lot of students. So let's go through that right now. So malaria, very common mosquito-borne disease in Southeast Asia regions. So we're talking like Malaysia, Philippines, those sorts of area. It is a disease caused by a protozoa, okay? Whereas dengue fever is a viral infection. I want you to know just how malaria is spread and just some basic statistics of malaria, okay? That's necessary. So, malaria obviously is spread through a mosquito and it goes through a life cycle, and symptoms of malaria include headache, pain, nausea, vomiting, and eventually death, okay? Some statistics here, again, you should remember a couple statistics just to back up your arguments. But if you are struggling to remember biology content, I would say you don't need to remember these, okay? Just it's good, but it's not necessary. All right. Next, we finish off the module with some culturally diverse strategies. So we're looking at previous methods of disease eradication and also some Aboriginal medicine. Okay, so historically, before we understood the causes of disease, before Robert Koch and Louis Pasteur, we didn't really know what caused diseases. We didn't know how they spread. We didn't know that pathogens were a thing. Okay, so how did we treat them before? Okay, so you do need to know this little part of the module as well. So let's look at the specific example of the Black Plague. Okay, the Black Plague or the Black Death was a really bad disease. It killed a lot of people in Europe. And we had these people known as plague doctors. Okay, these were what they wore. Plague doctors, they did contact tracing. They also did bloodletting. Okay, so some of the previous methods, you realize that some of them are effective, like contact tracing, wearing a long mask, using this stick to prevent close distances, but bloodletting was also something that they believed in. And bloodletting was just kind of cutting themselves, letting a lot of blood drain out, all of that nasty microbes draining out. And they thought that that could prevent the spread of disease and cure the infected person. We know now that that is very dangerous, okay, because we don't have a lot of blood in our bodies, and letting that blood out is not going to help our immune system at all. Um, they believe that diseases were caused by an imbalance of bodily humors. Okay, so blood, plaque, pus, those sorts of things, and they believe bloodletting will balance it. Okay, not the case. Drinking vinegar, I don't know how effective that is. I don't think it is at all. Bursting buboes was another thing. So a very common symptom of the Black Plague was they'll get these fluid-filled buboes, and they'll be quite big. And people back in the days they will burst it. Should you do that? Not really, okay? Because bursting the buboes just spreads all of that nasty liquid into the environment, allowing the pathogen to spread even quicker. So definitely not good. All right, so read through this, read through how we treated diseases before. All right, this part of the module, I really want you to remember. Indigenous medicine is something that they will test you every year. Why? Because they're required to add in Aboriginal components into your exam, and it's really important to recognize these Indigenous people. Okay, so just to finish off Module 7, we have Indigenous protocols, looking a little bit about intellectual property and what Indigenous people use to treat diseases. One example that I like to remember is tea tree. So tea tree is an example of a bush medicine that Indigenous Australian people use. They use Australian flora and fauna. The witchetty grub is a really good fauna to remember. Tea tree is a very good flora to remember. So tea tree, you might use it in your skincare, like in your serums. And what does tea tree do? It can be crushed and applied as a paste for skin. Really good antiseptic properties, which is why it is in your serums. Okay, tea tree cleans out or prevents the growth of bad bacteria and viruses on your skin and funguses on your skin. Therefore, it's in your serums, and that just prevents like acne development, okay, and pimple development. Another use of it is it can be brewed to treat sore throats. That is also a very good one. In this case study here, the Western Australian smoke bush, This case study very commonly referenced in trial papers and the HSC. Why? Because it is an application of indigenous medicine and also intellectual property. Okay, intellectual property just means property that you own that is knowledge. Okay, so you can actually put a patent on your intellectual property. But why is this case really important? It's because the Australian smoke bush, it was originally a bush medicine, it's still a bush medicine, it uses Australian flora, okay, but recently it has kind of potentially been known to be a potential treatment for HIV. If you don't know, HIV is a disease that is a really big problem right now. It causes AIDS and it is currently quite incurable. So, smoke bush has potential to cure HIV. Okay, potential meaning that there's no real backup for it yet. We can't release it out to the public, but there has been promising results. Okay, but it is intellectual property of Aboriginal people, so we should recognize them. But instead of that happening, a Victoria pharmaceutical company known as Amrad, okay, they bought a global license to patent smoke bush. That's a very big problem. Why? Because if this HIV treatment is successful, then all the money will go to Amrad instead of Aboriginal people. So that is going to be quite a big problem because it belongs to Aboriginal people. How can this big company just take out a patent? Because there's a lot of ethical issues related to intellectual property and not giving recognition to Indigenous people. There have been new rules and laws being developed by the Australian government, but it is still an ongoing process. So really know this example to back up your argument there. All right, so that is the end of Module 7. Okay, so go through our notes again, do a little revision. Module 7 is a hard module. Okay.